Hello, Theresa Stack, an associate professor at Montana Tech, and this is part two of the learning series, The Transmission and Determinants of Disease. And we're going to be speaking in the second half about infectivity, pathogenicity, virulence, disease transmission, and then finally wrap it up with some reporting systems or surveillance models that are out there. So an example of pathogenicity and virulence. Hepatitis A in children has a low pathogenicity and a low virulence. Many people may be infected, but the children remain asymptomatic. Pathogenicity, that means that they don't develop the symptoms, and very few develop severe illness or die. So hepatitis A, although it has a pretty decent infectability rate, has a low pathogenicity and a low virulence. Of course, with something like hepatitis A, if we are vaccinated against it, the pathogenicity goes down that much further. We can be infected by it, but our body will fight it off. Another example of pathogenicity and virulence, measles has a high pathogenicity but a low virulence. Almost all infected persons develop the characteristic rash and illness. So almost everybody who's infected has symptoms of the disease, but few develop the life-threatening pneumonia or encephalitis. Among persons with poor nutrition, poor health, measles is more virulent, with a mortality rate as high as 5 to 10%. So the infectability is dependent on the host and the environment, as well as the agent, right? An agent which dies off really quickly doesn't have a chance to spread itself. And also the pathogenicity has the characteristic of the agent, or can be determinant of the agent, the environment, and the person as well. And the virulence has some dependability on the host, the characteristic of the person, the characteristic of the disease itself, the agent, as well as the environment. So here are some cases, just some slides to look at comparing 2014 with 2015. And these are 2015 is when there was that spike in measles that occurred in the California area. Some more fun and exciting measles facts. The measles virus is highly contagious. Almost everybody who is unimmunized, who comes in contact with the virus, who is infected by it, will develop pathogenicity, will develop the signs and the symptoms of it. Among unimmunized people exposed to the virus, over 90% of them will contract the disease. The infected person is highly contagious for 4 days before the rash appears and 4 days afterwards. So you're infectious, not infective, you're infectious before you even realize that you have the disease. The measles virus can remain in the air and still capable of causing disease or pathogenicity in people for up to 2 hours after a person has left the room. So it has a pretty long shelf life there. So another example of pathogenicity and virulence, rabies is highly pathogenic and highly virulent. All infected individuals who are not treated with prophylactics to the clinical disease almost universally die. And there are some folks now, you can look at it on YouTube. I'll hopefully be able to play the video here, who've been put into a coma so that when the disease was at its worst and it had swelling of the brain because it causes an encephalitis, these people didn't have to combat the pain and discomfort of the disease in a waking state, and that subdued state allowed their body to fight off the disease. but it's very rare that somebody is able to survive a rabies bite without some form of intervention treatment. And as the public health director for Madison County, law enforcement and medical providers are required by law to report animal bites to us, and dogs without proper vaccination are quarantined up to 10 days. And this is true across the United States. So now we're kind of down to the end of our discussion here. We're going to talk just briefly about the chain of infection, different modes of transition, portals of entry into the human body, and finally wrap it up with a little herd immunity, and then, of course, talk about those surveillance systems. So, disease transmission, the chain of infection. Infection results from the interaction between our host, our environment, and our agent. The transmission of disease occurs when the agent leaves the reservoir or host and is conveyed through some mode of transmission. It enters the host, the susceptible host, the human, through some type of portal of entry. And this process is called the chain of infection, right? Wash your hands, wash your hands, wash your hands. The transmission of disease occurs when the agent leaves the reservoir. Bats are notorious for harboring diseases, all sorts of diseases which include, well, I'll just move on. Bats are notorious for harboring diseases. It's conveyed by some mode of transmission and enters a susceptible host. Bats bite people, or sometimes people come in contact with bat scat or bat guana. And so the disease leaves the bat and enters the human through some portal of entry, typically an injection from a bite, or it could be through ingestion if you had it on your hands and then you put your finger in your mouth or rubbed your eye, and that would be one way in which you can develop rabies. And so our reservoir, and again, could be a rat, could be the human, could be a water source. The agent is some form of microbe. It's transmitted to the human, and it enters through the eyes, the nose, the mouth, and through the digestive tract, the skin, or it could enter through other open ports in the bottom, yes, in the body. Well, that's a Freudian slip, such as the genitalia or the urinary tract. A reservoir is a habitat in which the infection agent normally lives. So a reservoir can be the environment, like a water reservoir, or it can be animals, insects, or humans. Animal reservoirs are notorious for causing diseases. If you look up zoonotics, you'll find a huge list of diseases that are brought to us by living close to animals. And so zoonotics are infection diseases that are transmittable from animals to humans, such as H5N1 and H7N9, which are the bird flus, as well as things like ringworm. Birds are reservoirs for West Nile virus and salmonella, as well as mosquitoes for West Nile virus, of course. Rodents are reservoirs for the plague, as well as typhus, and several mammals carry rabies, which includes bats and raccoons. So environmental reservoirs, plant, soil, water are examples of plant or environmental reservoirs, and this also includes our beloved Botox, which resides in the soil and is created from the same agent which causes botulism. So continuing our talk or our discussion on modes of transmission, we have one-on-one contact and non-direct contact. So for one-on-one contact, there are three different methods of a mode of transmission. The first one is direct, and that's simply body surface to body surface, such as shaking hands, kissing, or having sexual relations. So, for example, the common cold, STDs, and hookworm is transmitted this way. Indirect one-on-one contact would be the agent is depositing onto an object and it survives long enough that it can be transferred from one person to another person. So an example would be not washing your hands or using a contaminated instrument, or in the case of HIV, a contaminated needle. Some examples are norovirus, rhinovirus, and potentially influenza. So our third one-on-one mode of transmission is droplets, and this becomes confused with airborne. So the next slide will really make this clear for you. So droplets, contact is through the air, but the droplets need to be large enough and live for long enough for somebody to actually be contaminated that way. So for example, sneezing and coughing. So here is our example of Ebola. Ebola does spread through the air. So I can sneeze and Ebola would come out of my mouth and land on the table. But I wouldn't get Ebola from inhaling the air after somebody sneezed, unless of course I was so close that the droplets got in my mouth. But the droplets of the virus are so big that they fall to the floor, they fall to the surface so quickly that you become contaminated by then touching that surface and getting it in your body. Versus measles, which can be spread via airborne, and that's where the droplets are so small that they become aerolized and they can be transmitted through the air. They hang around there long enough that if you inhale, you can get them into your body. So our second form of a mode of transmission is non-contact. And airborne is transmission via aerosols that contain suspended organisms in droplets or in dust. And again, these have to be small enough to stay suspended. So TB, measles, chickenpox, smallpox, varicella is, which is chickenpox, is transmitted that way. And then the second non-contact form of transmission is a vehicle, and that's a single contaminated source that spreads the infection, such as the potato salad or cyanide poisoning in Tylenol. And this is where you're not in direct contact with the contaminant. The contaminant is contained in something else that you ingest, take in, or maybe if you stick your finger in your eye, you can get some kind of disease. So just to define that one more time, airborne transmissions occur through particles that are suspended in the air. Such transmissions occur over much longer distances than person-to-person droplet spread. So droplets are larger, they fall down much quicker. Aerosols can spread through the air and they become suspended. So a vector. Most vectors are arthropods of some form, such as mosquitoes, fleas, and ticks. They may carry the agent in their gut, and then the agent is deposited into the new host. But sometimes the vector actually plays a really complicated role. So the vector, say the mosquito, is contaminated by the agent, and then as that agent is inside of the arthropod's metabolism changes it, and then it becomes a infectious agent for us when the mosquito then bites us. So we are going to watch a short video on herd immunity.
How many people do you know who have had diseases like diphtheria, tetanus, polio, measles, mumps, or smallpox? If you live in the developed world, you probably don't know very many, which is a good thing because those diseases are not only unpleasant but potentially very deadly. And the reason that they are rather rare, if not eradicated entirely in the case of smallpox, is because of vaccines and the power of herd immunity. But what is herd immunity? And how do vaccines figure into it? Let's answer these questions using gummy bears. The very simplest epidemiological model, the SIR model, makes use of three categories: susceptible, the white gummy bears; infected, the red gummy bears; and recovered, the green gummy bears. Susceptible bears can get the disease, infected bears have the disease and can transmit it to susceptible bears, and recovered bears have had the disease so they can't get it again or transmit it to other bears. So here's our model. We have a population of white susceptible gummy bears, and we introduce an infectious disease. It spreads through the population of susceptible bears and infects them, turning them red. Some bears will die of the disease and disappear from the population, but the ones that don't will develop immunity and move into the green recovered category. The green recovered gummy bears are important because in addition to perpetuating the gummy bear population, they're able to interact with the red infected bears without any risk of becoming infected themselves. Assuming that all the gummy bears in our population are going to interact with a certain number of other bears one way or another, it's better for the overall population for red bears to interact with green bears rather than white bears, since the white bears are susceptible and the green bears aren't. As the number of green bears in the population increases, the chances of a red bear meeting a white bear go down until there aren't enough white bears left to sustain the disease in the population. Vaccines are really useful because they allow us to increase the number of green bears in the population without anyone having to be infected first. We can move them straight from the white susceptible category into the green recovered or immune category. No infection necessary. And if we can vaccinate a large enough proportion of the population, then we can prevent the disease from ever taking hold in the first place, and even the susceptible white bears will be protected from the disease, simply because they will never encounter it. The protection that a high number of vaccinated green bears provides to the small number of susceptible white bears is called herd immunity. Simply put, if enough of your herd is immune to a disease, it's unlikely that you'll get it, even if a few people get sick, simply because infectious individuals are unlikely to encounter susceptible individuals. This is why vaccines are important for public health, and why we don't see many cases of measles, mumps, polio, or smallpox.
So the video explained really well the concept of herd immunity, and there's are those among us who are not in favor of vaccines, and that is understandable, but I think you can understand by watching the video how it is a societal issue to the greater good for the largest proportion of the population to be vaccinated. And quite honestly, as my very good friend, the physician, said, sometimes the cure, although it may be not always the best option, it might hurt some people. It is certainly much worse than the disease itself. So the side effects from having a vaccine are much less complicated than the long-term effects of having a disease such as measles. And we're going to describe, now we're moving forward to describe our disease in terms of pandemic, endemic, and epidemic. And Ebola was an epidemic. An endemic, as graphically illustrated in the next slide, is when a disease occurs at normal levels. It is said to be endemic. So endemic is potentially the amount of diabetes that you have in a population. If it's stable, it's said to be endemic. A large spike of disease, more than the background level, is an epidemic. This may also be referred to as an outbreak, an epidemic. So here you can see your epidemic. A pandemic is an epidemic that crosses the globe. So Ebola had the potential to become a pandemic, and although it did cross into the United States and into some other countries, it was not at epidemic levels in those countries, and therefore it did not become a pandemic. So this is a really neat slide, quite old, but it shows an example of a pandemic, and this was from the influenza pandemic. And you can see that, I know it's a little blurry, the slide is in the book, I believe, but the different lines on the graph represent different countries, and they were all at a endemic level, which is a baseline level, and then it peaked around the globe entirely at the same time as most influenza does in this time period between September and usually March. So we're going to spend a little time looking at some epidemiological curves so you can understand these patterns of transmission and how epidemiologists plot them to be able to use them to understand what kind of an outbreak there is. So this is an example of a point source outbreak. So there was a point exposure with no propagation, and we can see that because the number of cases grew, peaked, and then came back down again. All cases appear to occur within one incubation period, suggesting that the cases did not arise from person-to-person contact. The fact that the outbreak was of a short duration suggests that it was a single brief, hence the word point, exposure that did not persist over time. Examples include that embarrassing diarrhea saga following a neighbor's summer barbecue, or a respiratory illness in workers followed by a breakdown of a fume hood. Another example of a point source outbreak. With continued source outbreak, so you can see that you have an incubation period and ebbs and flows, and it extends over a long period of time. As with the point source outbreak, a group of people are exposed to a single influence or agent, but here the exposure continues over a longer period of time. For example, a contaminated water source that doesn't get fixed, so the outbreak persists over a longer period of time. The relatively abrupt beginning of the outbreak suggests that many people were exposed simultaneously rather than it spreading by transmission from one person to another, because that takes some time. The fact that no cases arise beyond the incubation period following the termination of the exposure also supports this theory. So if you contrast few people to many people and then it dies off versus lots of people and lots of more people and then it dies off abruptly. Again, another example. And this is an example of an intermittent outbreak. You may occasionally see this pattern. This seems to be common in sources that are not well controlled, such as influenza. So our outbreaks reoccur. Depending on the time frame, it could be seasonal or weather-related, or perhaps due to a common source, such as an industrial contaminant being emitted at intervals. The gaps between the outbreak might initially suggest person-to-person transmission, followed by an incubation period. But this is unlikely because in a transmittable disease, the successive peaks would become larger and merge together. So that would be from a common source, and here's some examples of transmission due to person-to-person contact. Here, a single index case, for example, a returning traveler, infects other people, and cases arise after the incubation period. The outbreak wanes when the infected people no longer transmit the disease to one another. The graph suggests here that this was done rather quickly. Controlling the source is no longer sufficient to control the outbreak. So here is an example of a propagated spread, as we saw with Ebola. This begins with an infected person, the index case, but then develops into a full-blown epidemic with secondary cases infecting new people, who in turn serve as sources for yet other cases. This produces successfully taller peaks, initially separated by one incubation period, but the peaks tend to merge into waves with increasing number of cases in each generation of the disease. The epidemic continues until the remaining number of susceptible individuals declines, or until intervention measures can be put into place. So this graph was showing the propagation of Ebola from the initial and from the initial, excuse me, epidemic stages on July 13th, and how quickly it grew, and it started to multiply itself as more and more people got it, more and more people were able to spread it. And it was projected at that time, or forecasted, to go quite severe. Other time considerations with our propagation curves. Is the frequency stable over time, or is it changing? That's endemic versus epidemic. Are there seasonal fluctuations, summer versus winter or spring versus fall? And is there a cyclic variation? Measles epidemics tend to occur every 2 to 3 years, influenza A every 2 to 3 years, and influenza B every 4 to 6 years. And our last section is just a brief discussion on notifiable conditions, what some of them are and what the requirements are. In the public health department, we had a defined list of items that we would report out our numbers to back to the state, who would filter that back to the CDC. We did it every week, and then they returned and told us how many cumulative cases were of a certain disease, such as maybe influenza, in Montana. So I was in Madison County. I would report my data along with Butte Silverbow. They combined the data, it would go to the state, and then we'd get it back, and then all the states, of course, would combine their data and would go to the CDC, and we'd be able to see in a picture what the whole United States looked like. There are several conditions that fall under the list of nationally notifiable diseases and conditions. These conditions get reported through the public health infrastructure. Because the system relies on reporting of cases by physicians and other healthcare providers rather than an active system, it's called a passive system. And I quite honestly, every Thursday, used to send a fax and then an email reminder out to all of our physicians and clinics and ask them if they had any reportable conditions, some of which you can see on the next slide. And some more notifiable conditions: chlamydia, gonorrhea, hepatitis, tuberculosis, pertussis, encephalitis, brucellosis, West Nile virus, Q virus, rubella, chickenpox, hepatitis D, malaria, rubella, and state-specific, we actually have rules in place in the state of Montana, and your state may be different, of course, that all animal bites, driving impairments, so that meant if an individual had seizures and it might impair their driving, as well as gunshots needed to be reported to uh, the public health department, and then gunshots, of course, are reportable to the police department. So that is the end of a very long section of slides. It's why it was broken out into two categories. If you feel yet somewhat confused, your highlights are to recognize who, what, why, when, how, and where, the epidemiological triad, and how the characteristics of the host, the agent, and the disease come together. Also, to recognize that there are different stages of a disease: susceptibility, this subclinical stage in which you may be you may be contagious even though you don't have outward signs, and it's not until you have the outward signs and you're clinically diagnosed that that becomes a marker in time, and that it's slightly different for diseases such as cancer versus communicable diseases. So here again is our timeline. We looked briefly at Typhoid Mary and her being a carrier. Definitely spent some time on our different modes of transmission, as well as direct versus indirect contact. Finally, we took a look at some epidemiological curves just so you can have an understanding that diseases present themselves in different manners within a population. And finally, we ended with reportable conditions. I hope you have a great day.